All right. So we're gonna start the workshop now. So hi everyone, thanks for coming and joining our first academic workshop. As all of the flyers mentioned, we're gonna be doing an exploratory data analysis workshop. And so today will just be an introduction, introducing what is exploratory data analysis. Yeah, I can actually go to the agenda. But yeah, just talking about what is data exploratory analysis, kind of like the workflow that you'll go through, and the specific data set that we'll be working on in this specific workshop are microarrays. So we'll do an introduction to that, the typical microarray analysis pipeline, and then the data set that we'll be working with. So hopping right in, what is exploratory data analysis, right? So this is a technique that involves the analysis, the manipulation, and the visualization of your data set to uncover meaningful information, right? You're trying to figure out what patterns you can identify in the data set. So to kind of better understand this concept, let's say that there's this data set on student performance. So the data set has the following attributes. There's student ID, student name, gender, graduation year, family size, parental status, the parent's level of education, attendance rate, and GPA. So taking these attributes, I'm going to open this up to you guys, but what questions can we ask to gain insights about student performance? Anyone want to raise hands? Yeah, so we could see, maybe is there any correlation between attendance rate and GPA, right? Anything else? Yeah, that's definitely doable too. And then we can even just do basic descriptive stats, right? We can see, oh, how many students are graduating in so-and-so year, so class sizes, right? We can see maybe what percent of students have an attendance rate that is greater than 90% or something like that. So that's kind of the idea behind data exploratory analysis. You have a huge data set, you're trying to figure out what type of patterns you can unravel from it. So the basic workflow, you have your raw data. So this could be data that you downloaded from a public database, data that you yourself created from sampling, so on and so forth. You have your raw data. And the number one step you have to do is pre-processing. You'll typically have, your data will be noisy most of the time. And there's a really common phrase that you may hear in your data analysis classes, which is garbage in, garbage out. So if your data has really bad reads, if there's a lot of noise, then the analysis that you gain from that will also may not be the best, may not be the most accurate. And you don't want your results to be influenced by bad data. So this is why we go through pre-processing. So first you clean the data, you're removing any noise, any of those bad reads, any missing reads, and any redundancies. So that's what's happening during data cleaning. Then there's data reduction. So typically the data that we work with, big data is a big thing nowadays. We will have data sets with multiple features, right? Multiple columns. And so during data reduction, you're trying to condense this data set into more simpler forms. So a common algorithm is linear dimensional reduction. And this is basically, if you guys don't know, let's say you have height and weight in one dimension, right? You have a table, height and weight. And so that's one dimension. And typically in our data sets, we have multiple different features. So that might be height, weight, age, I don't know, something, diet, I don't know, something like that, right? And so that's multiple features. So now you're looking at 4D, 5D, 60, 1000D, I don't know, something like that. And that it can easily get very complex. And so during linear dimension reduction or data reduction, you're trying to condense this, you're trying to make it more simpler so you can identify patterns. Did you have a question?
Yeah, it's just cutting out the columns you don't need.
Yeah, so that goes under data cleaning.
Okay.
So data reduction, there's a specific algorithm. If you want to look up linear dimension reduction, that's a good topic, but it's like a specific algorithm, and it'll basically condense the data into lesser dimensions through certain mathematical operations. But there's that. And then data transformation is when you're starting to think about how you can organize this data in a way that you can extract these patterns from. So that could mean anything from perhaps you have cells that are unstructured and you want to convert that to structured data. Or you may have two variables that potentially could be combined into one. So you're thinking about how you can reorganize the data. And then finally, there's normalization, where you're trying to make sure all your variables are in one scale. So for example, you could have age and height, right? And that's two different scales. You're looking at years and you're looking at, I don't know, maybe centimeters or inches. And so there are two different scales and you want to put that into one scale. So typically you'll look at it from 0 to 1, and there's also mathematical algorithms for that you could do. But yeah, that pretty much encompasses the pre-processing steps. And once you complete these, your data is ready for analysis. So from there, you can do statistical analysis. And statistical analysis is when you're doing statistics to observe any trends. So that could, I know I mentioned before, descriptive stats, mean, median, standard deviation, variance, such like that. You could do correlation. You could do statistical significance tests, so t-test, ANOVA, such like that. And we all know that looking at numbers is more difficult to interpret than visuals, right? So that's where graphical analysis comes from. So this may be heat maps or bar graphs, box plots, so on and so forth. Yeah.
Something. So I think one thing that I've learned after working in a serious lab is that most of your time spent is going to be in the pre-processing.
Yeah.
Like you're not going to be doing too much statistics. Like you will do statistical analysis, but you need to clean it very well. You need to make sure all of the pre-processing steps, and all of these will be online because people have made a standard way to do them. So you'll follow it online. Like you'll find a paper that's like, we do it this way, we do it this way. You pretty much will be following those steps, and you might change a few parameters, but yeah, I would say, and Sahiti can agree, like most of your time is spent just cleaning the data, making sure it's good before you analyze it.
Yeah, so, and that again goes along with garbage in, garbage out, because you have to make sure your data is of good quality before you can do any analysis on it. Yeah, go ahead.
Ask a question about two questions. Are you going to be talking about data reduction a little bit further?
No, I will not. But if you'd like me to explain it more, I can go to the board.
Because I'm kind of curious, but I don't know if that suits you.
Yeah, I mean, would you? I don't know the algorithm from the top of my head, but I know the concept behind it.
Okay.
So let's say.
The second question.
Yeah, go ahead.
The 4 steps that you outlined in data processing, are they done in order? Or do you
I don't think,
and then you do them over again? Or?
So normally normalization is the last step. That's what you do after, you got your quality values. But I believe data cleaning and data transformation, I guess they kind of, I don't know, I don't think it really matters. But I do think that data reduction and normalization, they're more like near the end. And there's definitely more steps during pre-processing too. It definitely depends on how your data set looks like.
Yeah, these are very broad steps. Like data cleaning, there's like 5 steps. Like there can be a lot of steps within data cleaning, and there can be a lot of steps within data reduction. But those are like the broad, like steps. And they don't have to be in that order, but that is like the general order that. Anyways.
Yeah.
Say I haven't done minor stats. Sometimes when you get to the stats, you'll notice something wrong with the data you noticed before. Then
you need to go back.
Additional transformations.
Yeah, so it's always a tricky place to be with pre-processing. So going on with minor reduction, right? So let's say you have a data table. So I'm saying height and weight, right? And you have some numbers. So on a plot, this looks like maybe something like this, right? And this is X and this is Y. But what if we introduce another variable. And this is age. So now you're looking at 3D. So instead of these values, it could look something more like this on a 3D plane. What if we add another variable, right? And it keeps going. So now we're looking at more and more variables, more and more axes. And in my head, 3D is already like kind of difficult. And I can't even imagine 4D. So even though computers are great and all, this gets complex very, very easily. And so during linear dimensionality reduction, I know in how many of you guys are bioinformatics majors? Okay, health informatics? Okay. I think this is pretty relevant to you guys too, because I know you guys work with data sets a lot. But I don't know how it is in health informatics, but in bioinformatics, there's two processes, PCA plots and UMAP. And so there's something called principal component analysis. If you guys want to research that, whoever's interested. But basically, you're taking all of these variables and you typically look at the first principal component and the second principal component because those are the two that kind of condense the data the best. And now you're looking at something like this. So this is PC1 and PC2. And you'll get, okay, this is, what's this? Height and weight, right? Or actually, no, let's look at disease and control. So you'll have height, weight, age, so on and so forth for disease, and you'll have the same for control. And so when you do linear dimension reduction, you could have clusters like this and like this. And these points will take all of these into consideration. And so this could be something like for healthy cells, and this could be for disease cells. And you can see that they're clustering in certain positions, and you can gain insights from this because you can see like, oh, there's actually differences in them. If that made sense.
It's like some binary and you will like take two components from this to one. Similarly with this.
Yeah, so actually when you do principal component analysis, you'll get another data frame with all of the principal components and so principal component 1 will have values and each value will coordinate with all of these values condensed into one value. So, and that's what you're plotting.
Can I also add on to the principal component?
yeah.
So to grab the idea of principal component, I can maybe try to word it a bit different. But can I draw on the board?
Yeah, of course, go ahead.
So there's this thing, an RNA sequence analysis, where you have a graph that looks like this, and it's going to be every single one of these. So PC 1, 2, 3, 4, all the way to the very end. So you'll see it, it'll look like this. Every single time, it'll look like this. And what this graph will show is the further out you go to these principal components, so this will be like principal component 20. The further out you go, the less valuable it is to you. Because this won't have much of an impact on your data. So part of data cleaning is you have to like consider, do I want to analyze PCA 20? Like not really, because this is going to cost money and time when you run your application on a supercomputer. So you have to decide like what PCA you want to stop at. So like here, maybe you have to rationalize it, but maybe you decide here PCA 5, like this. But what she's showing too.
What's on the y-axis?
What's that?
Do you want to talk about what's on the y-axis?
The y-axis. This is just like significance or weight, like how important it is in your data set. So as you go further across, it gets less important. So that's why when she shows this graph, PCA 1 and 2, these are always going to be, well, not always, but nothing's ever always, but most of the time, PCA 1 and 2 are going to be the most influential dimensions of your data. So that's why people do PCA 1 and 2.
Yeah, and if you guys are curious on how a PCA data frame looks like, I have some on my computer. I can show you guys after the workshop. But yeah. Any other questions before I move on? And also feel free to get coffee. And I'm just sitting there. So whenever you guys are ready. Yeah. All right, moving on. All right, so we talked about exploratory data analysis, and so now it's moving on to microarrays. So a microarray is basically a chip that contains thousands of these pores, and each pore contains a specific DNA sequence known as probes or oligos. And these hybrid I do want to mention that each probe has a unique type of DNA, and these DNAs typically cover a short segment of a of a gene. And so these are used to hybridize cDNAs, and I'm going to talk more about the experimental procedure in the next slide. But microarrays are typically or more commonly used for gene expression profiling. All right, so moving on to the procedure. In this figure, it's looking at two types of cells, a normal cell and a cancer cell. But of course, it could depend on your control and treatment. And so they're isolating the RNA from these cells. And one thing to note is that RNAs are very unstable. So to retain the information during this analysis, you will reverse transcribe it into cDNA. I know this is very biology related. So if anyone wants me to go over what some of these terms are, I'm happy to go over it. But yeah, so okay. Yeah.
Complementary.
Got it? Oh, okay. Yeah.
I didn't know.
No, you're good.
Lines are talking about CDN. So RNA is just one strand fragment. It can just like modify itself into like pairing up with its complementing pair within itself. So it will not stick to the probe. So it's not identified like whether like X gene is expressed or not. So you make it stable complement.
And actually, so going off what you said, so you'll have like the mRNA sequence, right? And you could have like maybe A, U, G, C, and C, G, A, U here. And even though this is one single strand, it can actually create something called a loop where these two bind together and you get something like this.
It can be other way.
Yeah, or it could, like there's multiple structures. Yeah. So that's why we reverse transcribe it into cDNA. And so these cDNAs, one from the normal and one from, or not one, but cDNA reads from the normal and from the cancer. They are labeled with two different fluorescent markers. It's called cyanine 3 and cyanine 5. And so these cDNAs will go through this hybridizing step where, again, in the previous here, the microarray chip has these probes, right? And so the cDNA, the specific fragment will bind to that probe, depending on which probe it is. And so after hybridization, the chip is scanned using two different light sources with two different wavelengths to identify the different colors. So in here is the green and the red, right? And so after scanning, an image is produced, and then the intensity of the light is extracted, and it basically tells us the amount of the samples that were binded to that probe in each pore. Do we have any questions? I know that was a lot. Do you have one?
The probes are sequences you want to prepare.
Yeah, so typically the probes will be a specific gene or just to make it simpler, I would say gene. Okay. Yeah, and each probe will have thousands of these probes in each pore.
Can I ask a question about web lab stuff? So how long does this process, like from start to finish, take?
I'm not sure.
Okay, no, I'm just wondering because I've never done this.
Yeah, it would be cool to see, okay. But yeah, no, I have no idea.
Those specifically, but honestly, some of these with the microarray technology now can only be like a few hours, depending on the size of your genome, of course.
Yeah.
Things like, smaller, like viruses and things, you can have it done in a few hours with high-throughput sequencing. Yeah, that's another thing. Like I know that microarrays are a little bit more older. I feel like I think that RNA-seq is a lot more popular now. But that's like a whole different workshop. Yeah.
All right, so from here, so we got the image and we extracted the intensity of the lights. And so this is something, this is an example of how it would look like. So this is a cDNA microarray image. And from here, we can do a step called feature extraction to get this raw data that we will be working with. So this is the microarray analysis pipeline. Again, we do feature extraction. So from the image, we're getting, let's go back. We're getting the position of the cells and then the signal intensity of the fluorescence. So again, we had that cyanide 3 and cyanide 5. So that's the raw data that we're working with. And again, with any type of data analysis, we do pre-processing. So that's what quality control is. We're trying to see if our reads are of good quality. And then from there, we do normalization to make sure that the values from each of the samples are of the same scale, 0 to 1, and there's different algorithms too. Okay, and then from there, we do differential expression analysis to make sure that the genes that are cDNA fragments were associated with are statistically significant. And from there, we can do biological interpretation, so clustering, gene set, pathway, network analysis. And I know this is a lot, but to break it down, going back to exploratory data analysis pipeline, we're basically looking at that raw data, we're pre-processing it, doing statistical analysis and graphical analysis. Okay, so just an introduction about the data set that we're working with. So obviously it's microarray data, but the specific, I guess I'll introduce that in the next slide, but here's just some biology before I get into what the data set is actually talking about. So satellite cells are a type of stem cells that originate in the muscle tissue, and it's involved in skeletal muscle growth and regeneration after an injury to the tissue. And so it's said that during the late fetal development period, so when the baby is like growing, or when the fetus is growing, it's these stem cells are, replicating and they're differentiating a lot so that they can grow that skeletal muscle and those tissues during development. However, adult cells are known as quiescent, which means that they're in this state of, they're in this resting state. And so when we're adults, we have more of these adult stem cells, and they can be stimulated or activated when it's stimulated. So for example, if there was an injury to tissue, then these satellite cells will be activated and it will promote muscle growth and regeneration. So the objective of, I guess, the experiment that we're doing, or the research that we're doing, is we're trying to perform genome-wide chronological expression profiling to identify the transcriptomic changes involved in early postnatal growth to acquisition of satellite cell quiescence. So I know that was a lot, but basically we're looking at if there's differences in the genes from that prenatal stage to the adult stage. And so the data was taken from this study. It's called Early Postnatal Expression Data from Mouse Skeletal Muscle Stem Cells. And basically, they isolated these muscle samples from mice, and there were 3 stages that they were looking at through microarrays. So there's P1, P12, and P28. And the P stands for postnatal days. So it's 1 day, 12 days, and 28 days. And they're trying to see if the genes or the expression of the genes are different as the satellite cell develops. That makes sense. Is everyone good? Is there any questions? No?
I just
Okay.
have a question. Are
Yeah.
we going to do like an actual workthrough of this on a laptop?
Yes.
Okay.
So yeah, that will go in sessions 2 and 3.
Okay.
Sessions 2 and 3 will be more hands-on with Python and R. But today I just wanted to give a introduction to what we will be working with, because I know there's a lot of biology, there's a lot of medicine related stuff in the slides, so yeah. But that is all I had for today. And like I said, next time on session 2, we'll be getting hands-on with microarray data. And this event will be on October 27th. Same room, same time. Yeah. Thanks for you guys for joining. Thank you.